Carly Waring, and I've been working for the past 3 weeks at the McNagny Lab at the Biomedical Research Center. I had the privilege of working with Dr. Michael Hughes, and we were studying podocalypsin function in the adult kidney. It'll be there in a second. Do you need the pointer? I can just, motion. Okay, so podocalypsin function in the adult kidney. So you might be wondering kind of what kidney function has to do with heart and stroke, but actually they're really closely related. So hypertension or high blood pressure is one of several known risk factors associated with heart attack and stroke, as I'm sure you all know, and the kidney is a key regulator of blood pressure. So kidney function and hypertension are really closely related. So if you have high blood pressure, that can lead to kidney failure, and if you have kidney failure, that can lead to high blood pressure. So the kidney helps to control blood pressure in many ways. One of the ways that it does this is by monitoring water and salt levels in the blood, as well as filtering out waste. So for filtration purposes, the nephron, as you can see there, is the functional part of the kidney. In a normal human kidney, there's about 1 million of these nephrons. So the filtration unit of the nephron is called the glomerulus, as you can see there. On the glomerulus, there's these specialized cells called podocytes, and these are important for blood filtration. They look like this. The podocyte consists of projections called major processes and foot processes that you can see kind of branching off there, and they wrap around the endothelial cells like that, and they form filtration slits that are essential to kidney function. A protein called podocalypsin is highly expressed on these podocytes. So we don't know exactly how podocalypsin works, but we do know that it is essential for the formation of the foot processes, as you can see there. The reason we know this is that because when mice don't have podocalypsin, they don't form foot processes at all, as you can see there, and they actually die at birth due to high blood pressure and lack of urine production. So because these mice are dying, it makes it really difficult to study the role of podocalypsin in adult kidneys. So to solve this problem, mice were engineered that we can actually induce the deletion of podo in when they're adults. So to do this, we need mice that have two things. First of all, they have to have LOXP flanking their podocalypsin gene, and then they also have to have something called Cre. When mice have both these things, we call them FLOX-FLOX-Cre positive mice. And in FLOX-FLOX-Cre positive mice, we can inject them with a drug called poly-IC, and it induces gene deletion. So essentially what this is doing is the poly-IC goes and it turns on the Cre, and then the Cre acts like scissors to cut out the podocalypsin from the genome. When mice don't have this Cre positive, or don't have Cre, we call them FLOX-FLOX-Cre negative mice. And these mice look the same, but they just don't have the scissors, so they aren't able to cut out the podocalypsin. So we use these mice as controls in our experiments that we do. So while I was at lab, I had two main objectives. The first objective was to determine if poly-IC administration to adult mice deletes podocalypsin in kidneys, and if that deletion is complete and permanent. And then our second objection was to determine if the successful pododeletion alters normal kidney function or high blood pressure. So before we started looking at organ samples and cutting mice open and stuff, we wanted kind of a less invasive way to see if the deletion process was working after we'd injected the mice with poly-IC. So we injected the mice with a drug called EPO. You might have heard about EPO on the media before. What we know is that when we give mice a high dose of EPO, it stimulates the production of red blood cells, which leads to podocalypsin expression. So what we thought was that it would show us if the deletion was successful in the erythroid progenitors, because if there were higher levels in the Cre negative mice than the Cre positive mice, because then that would show us that podocalypsin had been knocked out in the Cre positive mice. So after EPO injection, we took blood samples from some of our mice, and you can see here this blue stuff inside the box is podo expression. So you can see that our Cre positive mice, our Cre negative mice have lots of podo expression, whereas our Cre positive mice have barely any at all. So When we graph the results, we can see how dramatic it really is. And what this is telling us is that, yes, our deletion is working and Podoclixin deletion was successful in the blood. But we don't care about that so much. What we really care about is if the pododeletion was successful in the kidney. So the next thing that we did is we took kidney samples from 3 Cre positive mice and 3 Cre negative mice, and we looked at the Podoclixin transcript levels. So what we found is that in 2 of our 3 Cre positive mice, we had much lower levels of Podoclixin expressed, but in one of them, the levels were up close to that of the Cre negative mice, which are the ones that don't have the scissors to cut it away. So what this is telling us is kind of that our pododeletion was a success, but maybe it wasn't complete. So once we kind of concluded that deletion was in fact happening, the next step was to see if the deletion was affecting normal kidney function. So one way that you can look at kidney function and assess how it's going is you can look at protein and urea levels in the blood and urine. So in a normal functioning kidney, protein goes in the blood and urea goes in the urine. But when the kidney isn't functioning properly, leakage happens and urea shows up in the blood and protein shows up in the urine. So the first thing we did to check and see if these levels were normal is we looked at the urea concentration in blood. So when we first did it, the urea concentrations looked pretty normal and pretty the same, but we decided that we needed a control. So we injected a few mice with a drug called LPS, which induces acute kidney disease. So as you can see with the mice with kidney disease, they have really, really high levels of urea, much higher than that of our Cre positive or Cre negative mice. So this tells us that our urea levels are normal and we're seeing no signs of kidney failure in the blood, even though we looked and saw that Podoclixin was deleted there. So then the next thing we did is we looked at protein levels in the urine. So we took urine samples, which was not fun at all, and then we concentrated the urine and we ran it through an SDS page gel, which separates proteins based on their molecular weight. So after we did this, we stained the gel with something called Kamasi blue, and we looked at the bands. So as you can see, most of these bands look pretty uniform, but it's possible that one of the Cre positive mice has a little bit higher levels of a protein called methylalbumine. That's not really anything that we need to worry about because it's not a lot bigger to show anything really significant. But if you look here at the very bottom, you can see that there's uniformly smaller bands in the Cre positive mice, and there's bigger bands in the Cre negative mice. This is consistent with other gels that we ran too, so we know it's not just like some kind of thing. But we don't know what this protein is. We know that it's about 20 kilodaltons, but we don't know what that means or what that is, or that's something that we would have to look into in the future. So in conclusion, we saw that deletion of PODO was demonstrated in the erythroid progenitors, which would be in the blood. We saw that there was reduced PODO gene expression in the kidneys, but we saw that in Podoclixin flox Cre positive mice, they did not appear to have impaired kidney function. So some stuff that we could do in the future to look in more in our research is we could compare the Podoclixin protein expression in kidneys instead of just looking at the transcript levels. We could induce kidney disease in our Cre positive and Cre negative mice and monitor kidney function to determine if the Podoclixin is required for recovery after damage, because that's something that it could be instead of just for maintaining normal kidney function. We could delete PODO during gestation to determine when exactly expression is required for normal kidney development. And then also we could perform a thorough analysis of protein to urine to identify unknown bands in the Kamasi blue stain. So of course I have to thank the Heart and Stroke Foundation for everything, because without you none of this at all would have been possible. I have to thank Mike and Fran for being so awesome and teaching me everything and letting me try things and make mistakes, and it's because of that I learned so much. Thank you Dr. McNagny and the Biomedical Research Center for giving me the opportunity to work in such a beautiful lab around such amazing people. Thanks Brenda and James, Jeff and Laylee for all your support and everything that you've done. So, thank you.